Hi, it's Dr. Phoenix. Today we're going to be looking at human chromosomes, and we're going to be looking at how scientists look at the chromosomes and determine um, certain characteristics that are carried by individuals, whether there are too many or too few chromosomes, whether the individual is a male or a female, and so on. Now, biologists will take a picture of the chromosomes during mitosis, usually metaphase of mitosis, and they will group the pictures of the chromosomes together according to their size and their centromere location, and they'll pair them up into homologous pairs. This type of a picture and arrangement of the chromosomes is called a karyotype. A karyotype looks something like this. A lot of times you'll see them in black and white, but they look something like this, and you can see that the chromosomes are paired up according to size and centromere location, and humans will have 23 pair of chromosomes. Now, out of these 23 pair or 46 chromosomes, just 2 of them determine the sex of the individual. And these 2 chromosomes are called the sex chromosomes. And they are usually represented as the 23rd pair. This is a blow-up of the 23rd pair. If there are 2 what's known as X chromosomes, then the individual is a female. And if there are 2 of what's known as the if there is an X and a Y chromosome, then the individual would be a male. Now, the difference between the X and the Y chromosome is primarily size. The X chromosome is much larger than the Y chromosome, and so as a result it can carry many more genes on it than a Y chromosome can. So again, as we're looking at the karyotype, the 23rd pair are the sex chromosomes, and all the other chromosome pairs, or all the other chromosomes, are known as autosomes. Autosomes. So we have 22 pair or 44 autosomes and just 1 pair of sex chromosomes. So how do we determine the sex of an individual? How is that determined? Well, again, a female is an XX in terms of her sex chromosomes, and a male is an XY. So during gamete formation, we know that these chromosomes will separate, and in the male these chromosomes will separate, and if we fill in the Punnett square, we would see that there is an equal chance, a 50-50 chance, that the offspring will be a male down here or a female up here. But what is it that determined the sex of the offspring? You could see it's based on whether the male provides an X or a Y to the new zygote. All right, the female can only provide an X, so if a male provides the other X, then the new individual will be a female, but if the male provides a Y, then the new individual will be a male. Okay, but again, there's a 50-50 chance of the offspring being male or female. Now, there's some interesting hereditary situations that take place when genes are located on these sex chromosomes. And any gene that's located on these sex chromosomes are referred to as sex-linked genes. Sex-linked because the genes are associated with the sex of the individual. And there are many genetic disorders that have been shown to occur as a result of being carried on the sex chromosomes, particularly the X chromosome. Remember, the X chromosome is much larger than the Y. There's very few genes on the Y chromosome. And as we can see here, the Y is physically much smaller than the X chromosome and therefore carries many less genes as a result. Now, the thing that happens in sex-linked characteristics is this. we know that in order for a recessive trait to express itself, the individual has to be homozygous for the recessive trait. All right, we've seen that over and over again with the Mendel's problems that we've been working out. All right, there has to be two recessive alleles in order for the recessive trait to express itself. Now, there's going to be a little different scenario played out here in sex-linked because the male will only have many times one of these recessive alleles because it's not carried on the Y chromosome, because the Y chromosome is just so small it doesn't carry that particular allele. So we'll see that with some of these recessive genes that are carried on the X chromosome, the female will only express the characteristic if she is homozygous or has both of the recessive alleles, like we've seen over and over again. But the male can express the condition just by carrying one recessive allele. All right, so let's see how that plays out. Color blindness is an example of a sex-linked condition. And there are actually 3 human genes that are associated with color vision, and any one of these genes that are defective can cause color blindness, and they are carried on the X chromosome. So let's look at how this plays out. Let's say that the big C represents normal color vision and little c represents color blindness. Well, this is a dad that has normal color vision. The gene is on the X chromosome. There is no associated gene on the Y chromosome, again because the Y is too little. Here's a mom that's a carrier. She has a dominant normal color gene or allele for color vision and a recessive gene for color blindness. Now, she is not colorblind because she has a dominant normal gene, but she is considered a carrier. So during gamete formation, some of her eggs will have the gene for normal color vision, and some of her eggs will have the allele for color blindness. So let's see how this plays out. We can see that her daughters, potentially half of her daughters, could carry this allele for color blindness. Right? This particular daughter in this box would be completely normal, would have 2 normal alleles for color vision, but this daughter would have a recessive allele for color blindness. She would not be colorblind, however. She would just be a carrier like her mom. But look at the boys. This boy would be normal in terms of color vision, but this son would get the recessive allele from mom and would be colorblind as a result because there is no associated allele on the Y chromosome, and this son will be colorblind by just having one recessive allele. And that's the way sex-linked traits generally work. Most of the time the boys have the condition by just having the one recessive allele, and they got it from their mom. Okay, the girls very rarely have the condition because the only way they can have the condition would be if they got one of the recessive alleles from mom and the other would have to come from dad, which means dad would have to be colorblind. So in order for in order for a girl to have a recessive sex-linked condition, mom would have to be at least a carrier and dad would have to have the condition. Hemophilia is another sex-linked condition. This is a bleeding disorder. It is transmitted exactly the same way as we just described. Hemophiliacs are individuals that where their blood won't clot properly and they possibly could die from very minor cuts or bruises. Again, here is mom, who is a carrier but doesn't have the condition. She will pass on the condition to her sons, or at least 50% of her sons have the potential of developing hemophilia as a result of mom being a carrier. Her daughter could be perfectly normal or could also be a carrier, but she will not have the condition unless her dad actually has it as well. Muscular dystrophy also is a sex-linked disorder and is transmitted in exactly the same way as we just described. Muscular dystrophy results in severe muscle weakening that eventually results in the death, and it's usually boys that have this condition, and most of the time these boys will die sometime in their teenage years. All right, so in order for geneticists to determine whether or not these conditions are a result of being passed on by genes rather than coming from the environment or some other cause, they will study to see whether these traits are passed on from one generation to the next. And in order to do that, they'll use a chart called a pedigree chart. A pedigree chart. And a pedigree chart will basically show the relationship of these genetic disorders from generation to generation. In other words, who is getting them, mom or dad, and you can kind of figure out whether it's passed on as a result of a dominant gene or recessive gene, or whether it's sex-linked. And so genetic counselors can analyze these charts and determine the genotypes of family members. So let's just look at the basic key for a pedigree, and then we'll, in another video, look at how we can analyze pedigrees. All right, so this is a pedigree showing 1, 2, 3 generations. All right, a square represents a male and a circle represents a female. This line across these, the male and female, represents a marriage, and coming down off of that would be a vertical line and a bracket that would represent their children. So this mom and dad had 3 children, 1, 2, 3, 2 girls and 1 boy. Now, if a circle or a square is not shaded in, that means that that individual does not express whatever trait that is that we're looking at in this particular scenario. Again, that represents the marriage. If the circle or square is shaded in, that means that that individual expresses the trait. It could be colorblind, it could be hemophilia, whatever trait we're studying in this particular situation. All right, so before we go into different examples, I think we'll stop right here so we can focus on analyzing pedigrees in one particular video. All right, so I hope this is helpful to you. After you've viewed this video, go ahead and queue up the next video on analyzing pedigrees, and we'll see how biologists and genetic counselors can actually determine the genotypes of particular genetically transmitted disorders. All right. Again, have a great day. We'll see you in class.